In this video, we're going to be discussing local cell signaling mechanisms. And before we continue, please like and share the video and perhaps favorite it so that you can use it to study in the future. Now, when it comes to cell signaling, we can have cell signaling over a long distance, or we can have cell signaling within a tissue over short distances. Now, long distance signaling is usually mediated in the body by the endocrine system, and that's called endocrine signaling. And in endocrine signaling, you have some sort of cell that produces a signaling molecule called a hormone, which is then released into the bloodstream, travels in the bloodstream, and eventually exits the bloodstream at a different tissue and binds to receptors on target cells there, causing some sort of change in activity. And that's long distance. Local signaling, or short distance signaling, is classically divided into three types, okay? And so one of those types is going to be autocrine signaling. And then there's paracrine signaling, and then also something called direct signaling, aka this fancy name called Juxtacrine. Okay, this is the same thing. So what are these? What are the differences between them? So autocrine signaling is when you have some sort of cell and it releases a signaling molecule that will then bind receptors on its own membrane, causing some sort of change in its own activity. So the cell signals itself. That's autocrine signaling. Paracrine signaling is when you have a cell releasing some sort of signaling molecule or signaling factor, which will then stay in that same tissue, diffuse in the fluid of that tissue, and eventually bind to a receptor on another cell in that tissue, changing its activity in some way. And finally, juxtacrine signaling is when you have two cells in physical contact with each other, whereby there's a ligand of some sort on one of the cells and it binds a receptor on the other cell, they're in physical contact with each other, they're literally being held together in some way like that. And that causes some sort of activity change, typically in both of those cells, okay? That's juxtacrine signaling. And so notice that they're very distinct. Autocrine signaling signals itself, paracrine signaling is short distance, but they're not in physical contact, and juxtacrine signaling, they are in physical contact. So to solidify this concept, let's go ahead and do a practice question. The practice question is as follows. So researchers are investigating some sort of novel signaling molecule, which is called Nexlin X, which appears to play a role in liver regeneration following partial hepatectomy, which is basically where they remove a part of the liver. In an in vitro experiment, hepatocytes were cultured in a dish and stimulated with Nexlin X under varying conditions, and the following observations were made. So condition A, when hepatocytes were cultured at low density, Nexlin X failed to induce proliferation. Condition B, when hepatocytes were cultured at high density and in direct contact with each other, significant proliferation was observed. So here, we got to pay attention. They say direct contact with each other. And what local cell signaling mechanism does that? Well, that's got to be juxtacrine signaling. So we know that juxtacrine signaling has to be playing a role here, since it seems like when they're in direct contact, there's some sort of proliferation. Now, condition C, when high-density cultures were separated by a semi-permeable membrane, allowing diffusion of soluble factors, but preventing cell-cell contact, no proliferation occurred. So what were they doing here? Well, they were preventing physical contact, thus preventing juxtacrine signaling, but they were allowing paracrine signaling. But there was no proliferation, so there's no paracrine signaling ongoing. And then condition D, addition of anti-Nexlin X antibodies to high-density cultures blocked proliferation. And that really just shows us that this Nexlin X is important for this process, because when you block it, you don't have the proliferation anymore under the condition where you normally did have proliferation, which is when they were close together in physical contact, presumably exhibiting the juxtacrine signaling. So which of the following best describes the mode of signaling? Well, it's got to be juxtacrine signaling, and so that's going to be C.